Hello friends, welcome to TechQuest channel. This is for the medical technologist by Your Tech. In this video, we will discuss about bacterial identification methods using biochemical tests. Bacteria has many enzymes for their survival and metabolism. Some are microbial virulence factors. They provide microorganisms with the capacity to avoid host defenses and damage host cells, tissue, and organs in a number of ways. The examples are catalase and coagulase. Catalase is an enzyme catalyzed by decomposition of hydrogen peroxide to water and oxygen. Neutrophils and other phagocytic cells use peroxidase to kill bacteria. The enzyme NADPH oxidase generates superoxide within the phagosomes that are converted to hydrochlorous acid oxidizing agent kill phagocytosed pathogens. Catalase is a very important enzyme in protecting some of the bacterial cell from oxidative damage by reactive oxygen species. Bacteria can be classified into catalase positive and catalase negative for identification. Carbohydrate utilization. Bacteria are differentiated by the carbohydrate they metabolize and the types of quantities of acid produced. The gas produced from fermentation are a mixture of hydrogen and carbon dioxide formed through the cleavage of formic acid. Gas is detected using a carbohydrate fermentation broth into a small inverted Derham's tubes are placed. Even trace amount of gas collected as bubbles under the Derham's tubes are detected. The bacteria fermenting the sugar must have the enzyme to allow the sugar to enter the bacterial cell and the concerned enzyme to ferment that particular sugar. The bacterial fermentation of lactose is more complex. Lactose is a disaccharide composed of glucose and galactose connected through a glycolytic bond. To metabolize lactose, the bacteria should have three enzymes. The first one to allow the lactose to enter the bacteria. The second enzyme to break down the beta galactosidic bond to separate glucose and galactose. The third enzyme to ferment the glucose. Amino acid degradation. Identification of the ability of bacteria to produce enzymes that either deaminate, dihydrolase, or decarboxylase of certain amino acids are often used in identification schemes. The amino acid substrates must often tested are lysine, ornithine, arginine, and phenylalanine. Decarboxylases cleaves the carbox group from amino acid to form amines. Deamination of an amino acid phenylalanine occur in air. The presence of end product phenylpyruvic acid is detected by the addition of 10% ferric chloride that result in development of a green color. Indole test. Indole is generated by reduction deamination from tryptophan via the intermediate molecule indopyruvic acid. Tryptophanase catalyzes the deamination reaction of the amine group of the tryptophan molecule is removed. Final products of the reaction are indole, pyruvic acid, ammonium, and energy. The indole reagent containing para-dimethyl amino benzaldehyde reacts with the indole present in the medium, forms a red resinidol dye. Hydrogen sulfide production. The ability of certain bacterial species to liberate sulfur from sulfur-containing amino acids or other compounds in the form of H2S is an important characteristic for their identification. Aerobic and anaerobic. Some of the biochemical medium has slant on the bud. When inoculated, a stab in the bud and the streaking on the surface of the slant allows the bacteria to grow in aerobic on the slant and the bud for anaerobic conditions, so that bacterial ability to utilize the medium both conditions aerobic and anaerobic. Example, triple sugar ion medium. Few drops of sterile paraffin oil is added after inoculation of media can block air circulation that can provide anaerobic conditions for the fermentation identification. Example, oxidative fermentative media. pH indicators. pH indicator is a chemical detector for hydronium ions or hydrogen ions in the Arrhenius model. The pH indicators are mostly used in microbiology media and biochemical tests that can give the color change that are visually recorded. Mannitol motility medium. Mannitol is a type of sugar alcohol. Mannitol motility medium used to detect the ability of a bacteria to ferment mannite and produce nitrogen gas and to detect the motility of the organism. The medium contains low agar concentration for the motility of bacterium, phenol red as indicator at pH 7.6. When the bacteria ferments mannitol to change the medium into acidic, that changes the color from red to yellow. The presence of bubbles indicates the presence of gaseous nitrogen production. Triple sugar ion medium. TSI contains 3 carbohydrates at 0.1% of glucose, 1% of sucrose, and 1% of lactose. When sugar ferments, they change the color of the medium from reddish orange to yellow. As the amount of glucose is low, the pH drop is reverted back to alkaline by the amino acid utilization. Phenol red is the pH indicator. Sodium thiosulfate in the medium is reduced by some bacteria to hydrogen sulfide that reacts with the ferric ions in the medium to produce black insoluble precipitate. The butt and the slant provides aerobic and anaerobic conditions. Production of other gases are seen as air cracks in the medium. Citrate. Sodium citrate in the medium used as carbon source and the energy source. Ammonium hydrogen phosphate is the nitrogen source. Citrate permease enzyme allows the citrate to enter the bacterium. The citrate utilization test requires oxygen so that the test is done on the slant. The citrate is oxidized from bacteria and is extracted with the release of carbon dioxide from the medium. Sodium citrate combines with carbon dioxide to form an alkaline product, sodium carbonate. The pH indicator bromothymol blue changes the color from green to blue in alkaline. The citrate positive organisms are Enterobacter species. The negative is Escherichia coli. Urea hydrolysis test. Urease test is used to determine the ability of an organism to produce the enzyme urease that hydrolyzes urea. Hydrolysis of urea in the medium produces ammonia and carbon dioxide. The formation of ammonia makes the medium into alkali and the pH shift is detected by the pH indicator in the medium, phenol red, that changes from light orange to magenta color. Urease positive organisms are Proteus vulgaris. The negative are Escherichia coli. With the help of this basic mannitol motility medium, triple sugar ion medium, indol test, citrate and urease test, we can roughly identify many of the pathogenic organisms. For confirmation of these organisms, there are many biochemical tests available for individual sugars and amino acids that can clearly identify the species and each bacteria. Thank you. Thank you for watching.